Good morning. My name is Marshall McDaniel. I'm an assistant professor in the agronomy department at Iowa State University. We're out here near the BioCentury farm that's right behind me, and we're out here to talk about soil health. Okay, so right here we're in the middle of a long-term experiment. It's called the Marsden Farm Experiment. The primary investigator here is Matt Liebman. He's also in the agronomy department. I work with him. And what this experiment is testing is the long-term effects of diversified crop rotations. And there are many researchers besides me that work on it. I happen to be looking at the effects on soil health, but there are people looking at weed suppression, water quality, carbon sequestration, the economics of 4-year rotations versus a 2-year rotation. And this experiment was established in 2002, and it has 3 treatments. So there's the kind of business as usual corn soybean rotation that receives synthetic fertilizer based on soil tests. And we have an example of one of those plots behind me here. Right now there's corn planted in this plot that's to my right behind me. And then there are 2 other treatments. There's a corn, soybean, and then oat, red clover rotation, and that's rotated every 3 years. And then there's also a 4-year rotation that's corn, soybeans, oat, alfalfa in 1 year, oat as the companion crop, and then alfalfa in the 4th year, and then it goes back to corn. And so we know that there are a lot of benefits to yield, both the cash crops that we're looking at, but then also there are other benefits to soil health that we're trying to investigate in my research group. Soil health is all the rage these days. Everyone's talking about it, from researchers to farmers. And to me, one of the basics of soil health is monitoring change after a management practice. That's kind of the idea and underlying principle with the soil health movement and what people are so excited about. And there are a bunch of ways that we measure change in soils, and I'm going to talk about those today. The other thing that differentiates soil health from the terms before it, like soil quality and soil tilth, is the emphasis on soil biology, which you're going to hear a lot about today as well. And the underlying idea with that is that soils are a living, breathing organism almost, and we'll go over all the organisms that are in there and how they contribute to all the things we care about as humans, and how they can contribute to one of those things, which is growing productive crops and promoting conservation practices. All right, so soil health is defined as the continued capacity for soils to support animals, plants, and humans. And within that, we kind of think of soils as being able to perform beneficial services to humans, and we call these actually soil ecosystem services. So many of these you might be familiar with, so just providing and provisioning food, fiber, and fuel, so growing crops is why we're out here. That's one of the main ones that agronomists are thinking about, but also sequestering carbon, retaining water, providing a habitat for soil organisms, a structure or a base or foundation for buildings to be built on. Those are all the things that soils do for humans, and we call those soil ecosystem services. And I'm going to talk more about those today in more detail. But the main thing I want to talk about is that there are soil health tests out there, and many of these tests are performed by laboratories. So you can grab a soil sample from a field and send it to a laboratory, and they will give you back an analyses that's similar to a soil fertility test. But one of the main things that differentiates these commercial soil health tests from soil fertility tests is that they give you back maybe a score in many cases, and they're also measuring some aspect of soil biology. That's the thing that really drives them. And they also will charge you a little bit more for those. They're usually more expensive than just your basic soil fertility suite that gives you NPK and your other macro and micronutrients. And we're going to talk about those today. So as I mentioned before, I talked about commercial soil fertility tests, and those involve grabbing a soil sample. Usually we take a composite sample from about a 2 to 2.5 acre field or portions of a field, and we send it to a commercial lab. They send us back results. Those results are the nutrients extracted at that time and at that place that you collected that soil sample. So a good analogy to that is that it's a picture of what's going on in your soil over the year-round growing season. And if you want a more kind of full picture of what's going on in the nutrient dynamics and what the root is experiencing out in the field, I might recommend this, which these are called plant root simulators, is the product name made by Western Ag. And what these are their resin. They're ionic resin membranes that are just inside a plastic strip, like a garden stake. And I'll explain these strips a little bit here, these resin membranes. So this kind of semi-transparent orange portion of these has a membrane in it that is either positively or negatively charged. So this one that I have the plus on, it's positively charged, or it's negatively charged, so it picks up the positive cations. This one is negatively charged and picks up those anions in your soil like nitrate. That's important. So how these differ than just grabbing a soil sample and sending it to a soil testing lab is that you're getting almost a movie instead of a picture, right? So I mentioned that a soil test is a snapshot of what's going on. These, you take a soil knife, and I'm going to demonstrate this in a moment. You place it in the soil so that it's under the surface of the soil, and then you press the soil back up against it, and it's collecting nutrients over however long you leave it out. From usually they suggest 2 weeks to a month, and then these are collecting nutrients as they're released from the soil, and however long you leave it out there, it's measuring the soil nutrient supplying power. So ammonium, nitrate, so your major forms of nitrogen that's available to plants, and then all your other macro and micronutrients are picked up with these two ion exchange resin membranes called plant root simulators, because they pick up kind of a more of a fuller picture of what a root is picking up. So I don't think we need to get out too far, but this is probably good enough. So I've taken the anion one, and we usually pick a row. Again, these are supposed to represent plant roots. So there are, for research purposes, there are many different ways to do this. So sometimes, we'll put something around, maybe a PVC tube to exclude roots. But here, and what we often do in my research group is try to look at what the root is actually seeing. So I'm just going to take a soil knife and kind of create a nice crevice. I just put the soil knife all the way through the surface of the soil in between the soybean plants here, and then I take the soil probe and I put it up until about I have an inch above the surface. And you want to be consistent. If you're looking at any treatment effect or anything, the important thing is being consistent, and I usually leave them about the top of this about an inch out of the soil surface. And so that's what it looks like. And then to finish it off, I usually just take another stab at the soil and push the soil up against it so you have good contact. And over here, do the same thing. And so now you've got soil that's in good contact with the probe, and you leave it out. So in about 2 weeks, we'd come out and collect this. So we just talked about a different way as opposed to traditional commercial soil tests to measure nutrient supplying power, which is a soil ecosystem service. Another soil ecosystem service that we kind of attribute to soil health is leaching of nutrients. And so we actually want to leach less nutrients. A healthy soil should supply plenty of nutrients to a growing plant, but leach very little of it. And in my research lab, we use another kind of simple technology that uses ion exchange resin, but beads instead of membranes. So the way this works is, you could see this is PVC. You could buy most of this at your local Lowe's or or Tysons or Menards. It's just a PVC coupling with two pieces of 2.5-inch PVC and or 2-inch PVC and nylon mesh holding beads in this nylon mesh. And just like those ion exchange resin membranes that are on the plant root simulators, these ion exchange beads are positively and negatively charged beads. And what those beads do is they collect both the anions and cations that are leached through a soil profile when we bury this in the soil. So the way this works is we either have to come out with a shovel or we have to come out with a backhoe and somehow dig a soil pit. And then on the wall of the soil pit, we dig a tunnel that goes under the soil surface. So we're kind of under a profile of undisturbed soil. And then we bury this resin lysimeter, and we backfill that side tunnel and then fill the main tunnel. And then we leave these out typically a whole year. And over that year, soil water's infiltrating and then leaching through the soil profile and through these resin beads that are collecting both cation and anions. There are other ways to measure how much nutrients are in the soil water or being moved through soil water. And one of those are what's called a suction lysimeter. And there's one of these at the edge of this plot here. And this has just a cap on it. And then there's some corrugated plastic. And then inside here, there's tubing. And this tubing, we put a vacuum on. And then 24 hours later, we can come out and collect soil water from a porous cup buried in this field at about 4 feet deep. And that tells us, how much of the nutrients that the crops aren't picking up, but that's being leached potentially to groundwater and then maybe to surface water. So this is how we tie soil health into water quality. So as I mentioned, soil health is all the rage, and many farmers and researchers alike are trying to measure changes in soil health after you make a management change, right? So the idea is tracking change in soil health because you want to know that you're getting towards a direction of improvement, right? So you're improving those soil ecosystem services like nutrient supplying power, the ability to provide nutrients to your crop and grow a better crop, reduce nutrient loss, and or maybe even sequester carbon. There's a lot of carbon market talk now, and farmers are interested in doing practices that sequester carbon. So soil health kind of encompasses all these benefits that we want to get out of our soils. And one of the most sensitive things, and seems to be a good indicator of soil health, is the soil biology. And we have this 2-year crop rotation behind me, corn and soybean rotation to my right, but to my left we have the 4-year corn, soybean, oat, alfalfa that also receives manure. And on average, the microbial biomass is between 20 and 50% greater during the entire growing year. And so what that means is that there's more fungi, bacteria, more biota that are churning over, increasing nutrient supplying power, likely sequestering more carbon than a 2-year corn soybean rotation that has less soil biology. And that 20 to 50% increase we measure with a method in my lab that's rather expensive and a little more technical. But we've been working with farmers on trying to measure that biological activity in soils by burying things that you could buy at your local grocery store. So things like tongue depressors or popsicle sticks that are made out of birch wood. We've been burying these out in soils, and then we'll retrieve them after a couple months to look at how much of this is decomposed in the soil, or even tea bags. So tea bags you can pick up at your local Walmart or any grocery store. You could typically find tea bags. And so we've been burying these out in soils and then coming out and retrieve them as a DIY or do-it-yourself indicator of that biology and activity in soils that is so important to soil health. We can use different varieties of tea, and it's good to know what type of tea. Some tea actually decomposes a little faster than others. This is a rooibos or red tea, and it has a higher carbon to nitrogen ratio. The higher the carbon to nitrogen ratio is, the slower the tea will typically decompose. And we think that soil that's really healthy should decompose something that's a little more tough on average to break down. It'll decompose it even faster. So what we do is we come out and usually about a third of the way in between two corn rows, we just dig a small hole about 3 inches deep. We place the tea bag into the hole and then just gently bury it. So after we bury this, we can mark it with a flag and come out and retrieve it in about 2 weeks. Is enough time for decomposition to occur. With green tea, which has a lower carbon to nitrogen ratio, decomposes a little faster. You might want to get out here in about 4 or 7 days, so just about a week. One of the keystones to soil health is the living organisms in the soil that drive a lot of the soil ecosystem services, or the benefits that human beings and plants and animals derive out of soils. And one of the main findings that one of my graduate students found was that the microbial biomass, or just the mass of the amount of biology under the 4-year rotation, which is to my left, compared to the 2-year corn-soybean rotation, is at least 25% greater, and that's consistent year-round. Almost the entire growing season, there's more microbial biomass under these soils under the diverse 4-year rotation compared to the 2-year rotation.